Hello and good morning. We are going to work this morning on genome alignment. Actually, it's alignment and analysis. So, we want to achieve three tasks here. So, we are going to map the reads to a reference genome. Then, secondly, we're going to use annotation to count the number of reads that are mapped to each genome. And then the third one, we're going to report the three genes with the highest number of mapped reads. So, you talk about what are reads, what are reference genome, what are annotations. So there are four data sets for this. So by the way, this is actually bioinformatics or computational biology. So the idea of applying computation to biological data, in this case genes. So the first data set we have is the complete genome for the E. coli strain K12 substrain MG1655, which is this data, genome.fa, this data set. Then the second one is the complete annotation in the GT format for this substring, the file with the .gtf extension, this file. So this is the file, the GTF, the annotation we're going to make use of. And then the pre-computed bow tie 2 index for this genome, this file, we're going to extract this file later on. And then a set of reads that we're going to use to um, map to the reference genome. We're also still going to extract it. So, so let's begin with what we're going to do. So we are told that we are going to map the reads to the reference genome. So the genome we are talking of is this genome. You can actually see it here. To see the file head genome.fa. The head version, just the first few lines of the file. This is the head genome. So, but what we are going to do, we are going to map these reads to the reference genome. This reads to this reference genome. So, but before we do that, we are going to index this genome. So, after indexing it with the bow tie, then we are going to do the mapping of the reads. So, we already installed bow tie. We already installed it. So, we run it to see whether it's here. So, it's here. So, we're going to use this file. It's a bioinformatics library used for indexing of the genome and mapping of also of the reads. So, but before we do that, we are going to extract the bow tie to index file, which we are going to use in the indexing of the genome file, genome.fa. So to extract it, we run this command bow tie to.bu, then the file genome.fa. No, we first of all extract it like I said. So we're going to use the extraction zf. So we're going to extract this, and it's going to be extracted into this folder. So we extracted it and it's already applied here, the extraction. Then we're going to run that indexing of the genome. So the indexing is this command we're going to use in the indexing of it, of the genome.fa. And then where we're going to store our index, the file that has been indexed, the name of the place we're going to do the storing of it is, let us call it bow tie 2 index. So we run this command now. It does the indexing. So what it does now is to index the genome.fa file. So within less than 5 seconds, the index has finished. This is a lot of commands which we may not be able to explain in detail what it does, but what you just have to understand is that it has done the indexing of the genome.fa file. So after the indexing and the after the indexing, now we are going to now map the reads the reads to the reference genome. Look at the reads here. Reads 0_045 r1.fastq.tg. So we're going to extract it, and then we are going to do the mapping now. So let us extract it. zxbf reads. So we're extracting it into this same folder. So it has been extracted. Look at the file here. You're going to see the reads here. I think a lot of chromosomes. I'm not very adept with the biology. I'm coming into this problem from the computational or data science background. So, but these are the reads. I think there's the chromosomes, and these are the ATCG that we see in genomes. But anyway, so do the mapping of the reads now. We're going to use another file, another this thing which we have installed already. You're going to use no, but first of all, you're going to map the reads. So to map the reads now, we're using this command bowtie to then bowtie to index the indexed file, and then the reads the file, and then it's going to output the name of the output of the read of the mapping of the reads. Let us call it output.sam. So if you are going to explain this command now, so this is the command for the function for the mapping of the reads. This is the indexed file which we got from the first operation we did when we indexed genome file, genome.fa. These are the reads which we just extracted now. And then the output of the reads will now be output.sam. SAM is also a bioinformatics extension where we are going to store in the reads, the mapping of the reads. If you use the reads to map the indexed genome file. So we are going to run this command now. So we are mapping the reads currently. It will take a while. It will take a while. So while we are waiting for that, we are going to be looking at the third part. Well, actually, the second part. So we are still actually in the first one, the mapping of the reads. So we first did the indexing of the genome, then we did the mapping of the reads. So these are the two parts of the first one. So the second one should be the annotation, using the annotation to count the number of reads that are mapped to each gene. Mm. So it has finished mapping. The reads have been finished mapping. Okay. Then what we are going to do now is we are going to count the number of reads that are mapped to each genome. And for us to do that, we are going to use the function, the library, feature counts, and it's also installed already here. Feature counts. We're going to use it to be able to count the number of reads, and you use this function and then use our annotation, the annotation file we have here, equally.gtf, to be able to count the number of reads that are mapped to each genome. So the function, the command for that is feature counts here. So this is the annotation file which we are going to use, and then the output of the counting of the number of reads, that is this minus o, is we're going to call it gene.counts.txt. And then re- remember that we got the results from the mapping of the reads. It's the output count, output.sam. So we are using this function and this annotation to be able to count number of reads that are mapped to each genome from this output file. And what we are going to get, the final result we're going to get is going to be stored in gene counts.txt. So we run this command. Oh, it has not seen this. So this, I think this is where the problem is coming from. So both are to index minus u. So actually what I did was I wrote the access of the output sum in a small letter instead of capital letter. So we're talking about making of the reads. So but I've already corrected it now. So it has done mapped the reads to um, reference genome, and it has given us the output. Look at the output here, output sum. So after this, you're now going to do the mapping of the reads. No, the counting. So the counting of the number of reads that are mapped to each gene. So this is where we're going to use the feature counts, the annotation equally for GTF, and then the outputs, we call the output gene counts.txt. And then we're going to access this output sum. So what it does here is going to use this annotation and then this feature count to be able to count the number of reads that are mapped to each gene from this output.sum and give us our report in gene counts.txt. That's what it's going to do. This function is going to do now. So we run it. So it has already done it. So here it tells us that it's one sum file, output.sum. So the output file we are going to, he has already given us the output file for what we just ran now. So this is the summary. So there are no multi-mapping reads, there's no multi-overlapping reads. So the features 4564 and the chromosomes we have is only one. So it successfully assigned alignment 79.9%. So this is the result of the genes and the counting of the number of genes that we have mapped. So this is it here. So this is going to be like a tabular data set. These are the gene IDs. The type of gene is chromosomes. So we have the start and the end, the strand, then the length of the genes. Then this output of SAM is the column that tells us the number of mapped reads. So for example, the gene ID of B0001 has the number of mapped genes 0. There was no mapped gene. So 002 has the number of 9. So it has 9 the number of mapped threads for 0. B0003 is 9, and as the case would be. So these are the informations which we get from what we just did now. So but now, the last one we are going to do now is here. We are going to find, use a Python script here actually to report the genes that have the highest number of mapped reads. So you're going to create a Python script and pass in this text file and then get the information of the genes that have the highest number of the mapped reads. So we are going to run it here. This is actually a Python Jupyter notebook. So let's see how we are going to do that. Mm, okay. I think I've already run it before. See, we have already run this function, so there's no need of repeating it again. So this is the file path, counts.txt. You use this. And so it reads this file, it reads this gene counts for this file path, and then these are the columns that we have from the gene file. Okay, you have to install this pandas.py and install import pandas, and then this, and then this. So we're going to rename this output.sum. Remember from what we got here, this is a column for the number of mapped genes. This output.sum. So we want to change it so that it can be read. The name will make sense in what we are doing. So we are changing it to count. So that's actually what we did here. We changed it, and then we are going to sort the value of the count column by in descending from the highest to the lowest. So this is the function that does the sorting of the values of the count of the gene column, the count column of the gene counts. So it does that. So it will this function. So look at it here now. So if you run like sorted genes, gene counts dot head, so it gives us the first 5. So this one is the highest, followed by this one and this one. This is the first 5. So, but what we need here is just the first 3. 1, 2, 3. So that's what we did here. So they get the 3 top genes. So the gene counts of the head 3. So this is the top 3 genes. The top 3 genes. B4466, B4696, and B197. So this runs and gives us the value. So the top 3 genes with the highest number is this. So this is just basically a very simple bioinformatics operation. So remember what we did here. From here, we started with here. We mapped the reads to the reference genome, right? And after that, we now did this thing. So after indexing of the genome, we mapped the reads and we got our output with SAM. Then we now use the annotation, this annotation equal to GTF to count the number of reads that are mapped to each genome. And we get our output from this function as gene counts or txt. And then the remaining, the last part was to report the genes with the highest number of mapped reads, which we did here, and gave us the gene ID and the number of counts, starting with B4466 with the highest count 22, and the last B1978, which is the third highest, which is 17. So this is just a simple operation of the alignment of sequence reads to reference genome and analysis of the results using a genome annotation. So maybe next time we delve deeper into other sections of bioinformatics to even see the areas we talk about, the genes, the exons, the chromosomes. What is a little bit deeper. So it requires people that have some subject matter expertise, or if like us coming from data science or AI, to read deeply into this field and see how we are going to apply computation into it. So if you have any feedback or any suggestion, you can put it in the comment section. Yeah, thank you very much.